These are the crickets that we would have been working with if we'd been able to stay here at Siena. And what I'm going to do is I'm going to show you how I can inject a cricket with 5 microliters of this red amaranth dye solution. Okay, so what I've got here is a short-winged female. So she's got an ovipositor here to lay eggs, but you don't see her rear flight wings sticking out at all. So I'm going to go ahead and I'm going to give her this 5 microliter injection. I have to brace my arm against something stable to do this. So I'm going to insert the needle between a couple body segments here in her abdomen, and then I'm just going to slowly inject in this amaranth solution. She's trying to be really disgusting by spitting out some of her stomach contents right now. Okay, so there she is. There she is. She's been injected, and then we'll wait and come back and take some blood samples. Okay, so what I've got here is a long-winged female. So you can see her ovipositor right here, and then her rear flight wings are actually sticking out back here behind her wing covers. So I'm going to go ahead, I'm going to give her an injection into the abdomen. I'm going to stick this between segments in her abdomen, and I'm going to just slowly add that 5 microliter volume of amaranth. You can actually see she's starting to turn a little bit pink as I add that in to her abdominal cavity. Okay, let's see if you can see that up close. There you are. Okay, so then for my next parlor trick, I'll poke the crickets again using one of these insect pins, and then I'll take a blood sample using one of these microcapillaries. This is similar to what you saw for taking crayfish hemolymph samples. Okay, so here again is one of the crickets that I injected earlier, and what I'm going to do is I'm going to take this pin and I'm going to poke her right at the base of her hind leg, and then if I hold her just right, I'll see a little bead of hemolymph appear there, and then I'm going to stick the microcapillary up against that to collect the hemolymph sample. Okay, you can actually see it's a little pink in color. So I've got a little bit of hemolymph. I'm just going to rock her around just a little bit to get a slightly larger sample there. I put the microcapillary up against that, so it's going to start collecting hemolymph. I'm going to need a little bit more. Let's see if I can get more. Maybe not in this case. Some crickets are juicier than others, so we'll call that good enough. Okay, and then I'm going to put this tip on here, cover the end, and then I'm going to squeeze it into one of these cuvettes to release that sample. Okay. Now we can measure the absorbance of that sample in a speck. Okay. Give her a poke, and very quickly there, a big drop of hemolymph has welled up. So I'm just going to put this microcapillary right up against that. It's going to get drawn in. It's not quite 10 microliters, so there's a little bit more. Just trying to touch just the tip of the microcap. You get even just a little bit more to well up. Okay, there we go. Put that cricket back, and then I'm going to take this bulb and I'm going to go ahead and I'm going to deposit that hemolymph into a cuvette. Okay. And give that a good mixing. And then we can go and we can read the absorbance of that sample in a spectrophotometer. Okay, for this next step, first I have to give you a warning. This is a little bit gruesome. What I'm going to be doing is I'm going to be very quickly euthanizing this cricket by decapitating it, and then I'm going to just show you how to prepare the dissection of this cricket so that we can examine its Malpighian tubules. So here I've got the cricket. I'm going to very quickly euthanize her by decapitating her. These iris scissors. Okay, and then I'm going to position her in this dissecting tray, and I'm just going to make a single cut along the ventral surface here. Okay, and then I will pin open her body cavity using this set of insect pins here. And in this case, it looks like this is actually a flight-capable cricket that has metabolically active flight muscles. I'll be pointing out some more details of her internal organs when I switch over to a slightly higher magnification. Alright, so now we're going to take a little tour of this cricket's digestive system and other internal organs. So I repositioned her a little bit. This right here is the cricket's crop. You can think of that as being like its stomach. And the whole digestive tract is going to be connected together continuously. So if I actually pull that up, I'll bring everything with it. And the Malpighian tubules here are associated with the hindgut. But I'm just going to move those out of the way for the moment and point out a couple other things. So there's a lot of glistening white fat all throughout her body cavity. Those are all parts of the cricket's fat body, which functions very similarly to the liver in mammals. And then you can also see her flight muscles here. So here's the set of dorsal longitudinal flight muscles running along what would have been her back. And then these are the dorsolateral flight muscles that run top to bottom in the cricket. And then she's a flight-capable cricket, so her ovarioles are not particularly developed, but her ovarioles, when they do develop, they're just right here. So they could be full of eggs under those circumstances. And you can see that the amaranth dye that I injected is distributed throughout the body cavity, but there's a stronger concentration of that close to the Malpighian tubules here. So what I'm going to do next is I'm going to prepare a wet mount slide of the Malpighian tubules, and I'm going to see if I can show you what those look like up close. I forgot I wanted to point out one more thing before I move on to that next step, which is it's also possible to see some of this cricket's tracheoles. They're these shiny white tubes running along the surfaces of the abdomen, and there's also a set of tracheoles that runs right along these flight muscles to help ensure that they're well supplied with oxygen during flight. Alright, so here's an even closer up view of the Malpighian tubules. So all this yellowish stuff here around the hindgut are the Malpighian tubules. They're kind of like spaghetti. And last but not least, here is a 100x magnification of the side of this Malpighian tubule. In this case, I didn't let this cricket sit for very long with the amaranth, so we don't really see much amaranth in association with this particular tubule. So let's take a look at another one and see if it looks any different. Here's just a view of a short-winged female's digestive tract while it's still more or less in place. You can see some tracheoles along the top, and you can see those yellow Malpighian tubules. Alright, if you look closely, you can actually see substances moving through these Malpighian tubules. So you can see active solute secretion happening. It's amazing. Again, a really awesome view of substances traveling through the Malpighian tubules here. So we're just dissected out of a cricket. Great stuff. Alright, if you now look at the lowest Malpighian tubule, you can see some red granules of that red amaranth dye. They're not coming into focus fantastically well, but they're there. We'll try and take a closer look at those. Okay, trying to take a closer look here. It's a little challenging because at a higher magnification, we're up to the 40x objective. We don't have as much depth of field, and I can't hold the camera and focus at the same time. So not a whole lot to see here at the moment. If we were live and watched for a few minutes, we'd probably see more. But there you have it. Cricket Malpighian tubules.